Friday, and that's what we do. 8:30, our seminars start today again with one of our heroes for so many of us, Dean Bok. I remember Dean since my early days in vision science when I was a student, and it was always, every single time, I have to say, inspirational to chat with Dean. And one characteristic thing about Dean that he never shy away to speak with students, that he always was extremely supportive. And this is perhaps true throughout his career to so many of us who learn of his really outstanding research over the years. And the other characteristic of Dean Bok is that he's continuously a contributor to vision field. So it's not like a series of fantastic papers and then nothing for a long time, but it is this continuum since 60s that really open up investigation of visual system, particularly retina RPE cells, to molecular investigation. So Dean, you couldn't make me happier for agreeing to come and present today. And with formal introduction, let's move on to Elliot.
Thank you, Chris.
Good morning, everyone. It's a great pleasure and honor for me to introduce today's speaker, Dr. Dean Bok. Dr. Bok received his PhD degree in anatomy at UCLA in 1968. Following graduation, he was immediately offered an assistant professor position at UCLA and became a full professor since 1976. He was awarded the Dolly Green Chair of Ophthalmology and distinguished the Research Professor of Ophthalmology and Neurobiology. Dr. Bok has been a pioneer in the field of visual science. His research group demonstrated that the retinal pigment epithelium, RPE, has a phagocytotic properties and contributes to the renewal of photoreceptors for the lifetime. Other major discoveries from Dr. Bok were the molecular and cellular mechanisms whereby vitamin A is transported from circulation into the RPE and how it is processed within the RPE. He also contributed to the understanding of the visual cycle and pathobiology of retinal disease. Dr. Bok has published a number of high-profile publications, including Science, Nature Genetics, Neuron, and JCI, and has published over 150 articles. He has won numerous awards and recognized for his mentorship as well. He has served on advisory boards for the NEI Research to Prevent Blindness, Foundation Fighting Blindness, and Macular Vision Research Foundation. With this, please welcome Dr. Bok.
Thank you very much. It's indeed a pleasure to join you this morning. Some of you from overseas, I understand, and I'm very grateful for this opportunity to share some things with you. I guess if we go back to the title, retinal pigment epithelial photoreceptor interactions, we could go all the way back if we wanted to 1876, when Franz Boll and Wilhelm Kuhne first studied the photochemistry of the retina. But I won't take you that far back. I will take you, though, to 1969 and share with the youngsters this ARO program on the left, which fit into my shirt pocket, 4 inches wide, 19 pages long, and that's compared to the program for 2 years ago, which was 488 pages. The paper that we presented at that meeting was entitled "The Role of the Pigment Epithelium in Rod Outer Segment Renewal." And what I'd like to do is remind the youngsters as to what that means, but then build upon the shoulders of the young folks who've worked on that project ever since. And I have been truly impressed by the fact that so much progress has indeed been made. This was my mentor at the time, Richard Young. Richard Young came to me one day while I was studying for one of his exams and said that he'd been studying the biosynthesis of the lens capsule, but he looked back to the neurosensory retina and noticed this wave of radioactivity and autoradiograms that moved over time toward the retinal pigment epithelium. And indeed, if he injected 2 doses of radioactive amino acid, he could see 2 waves of radioactivity. And he then went to the literature and noticed that a fellow by the name of Bernard Ruiz from France had published a paper already on this. And he decided he needed to do 2 things. First of all, Ruiz was learning how to do autoradiography at the EM level. And secondly, he clearly wanted to improve his histological methods. So he did that. And while Dick was in France, he gave me the opportunity to actually run his laboratory for a year, which was a wonderful experience. And we turned to the frog, interestingly, which is also the animal that Willy Kuhne and Franz Boll had worked on in 1876. And here we could see with wonderful resolution the band now in individual outer segments about 7 to 8 weeks after the frogs were injected with tritiated amino acids, approaching the retinal pigment epithelium. And then using EM autoradiography, you could see the whole series of events that unfolded. The labeling pattern represented what George Pallotti at that time called a multi-step process. George was studying these mechanisms in pancreas at the time, and we submitted the paper to the Journal of Molecular Biology. And it's the first and only time that an editor has asked me to submit more papers to the journal. He liked it because it went in hand nicely with his pancreatic work. So you see here radioactivity and after 15 minutes of injection over the rough ER, and then it migrates to what looked like to us like the Golgi apparatus, collected at the base of the outer segments, and then 8 weeks later in the frog, a packet of membranes was detached by the retinal pigment epithelium through a process called efferocytosis. We called it phagocytosis, but it's a process that's used by macrophages to quickly take out of commission dying cells or fragments of dying cells so that they don't leak their contents into the extracellular space and cause a lot of mischief like cytokine mobilization and so forth. At the same time, Yoram Heller, our colleague at the Jules Sinai Institute, was purifying rhodopsin for the first time. And he found that it was a glycoprotein rather than a lipoprotein. And we could follow the course of specific radioactivity in those rod photoreceptors. Over time, it reached a plateau, stayed there for weeks, and then precipitously dropped as the phagosomes were shed. Another discovery that was made from the EM level was that the labeling patterns in the rods was very different than that in the cones. The rods had a discrete band of radioactivity. Here you see it in the frog. Here in the absolutely giant rod photoreceptor of the mud puppy, which is 16 microns across. And in both animals, the labeling in the cone outer segments was diffuse. And that puzzled us for a bit, but then considering the morphology of cone outer segments and the fact that in most animals, at least, in most cones, the cone discs are actually open to the extracellular space. They're not always in regular register like this. Here in the goldfish, you can see that these discs are open right here. Here they're closed, here they're open again, but these aren't truly closed. The openings are not in perfect register and would be at perhaps right angles to the plane of the disc edges that you see here. So our interpretation is that this diffuse pattern is due to the fact that integral membrane proteins undergo translational diffusion in the plane of the lipid bilayer, thereby randomizing throughout the outer segment. The question though was, and we knew by this time from Steve Fisher's work, that these cones shed their discs just as rods do. So how in the world does a cone remain conical? It must undergo some extraordinary remodeling every day in order to remain conical. And to date, no one has really explored that with modern experimental techniques. We are very interested to see some of you youngsters getting into that subject. Now, George Pallotti described protein biosynthesis as a multi-step process, whereby things happen in the rough ER, the Golgi, and points beyond. And we wanted to study that process for rhodopsin. Where are the various groups of rhodopsin added in the cell? And we thought that would be a straightforward proposition, but it got a little bit complicated. By this time, David Papermaster and Barbara Schneider had done immunogold of frog inner segments and shown that using ferritin at the time as a label. Here, Alan Bird and John Flannery have used immunogold. The Golgi apparatus is heavily decorated with antibodies in the inner segment. So it became clear then that the rhodopsin was following a classical secretory pathway. And here, if we introduced various sugars, radioactive sugars, we saw that they were indeed incorporated in the region of the rough ER and the Golgi apparatus. So here you can see it better with N-acetylglucosamine. We know that glycosylation begins in the rough ER and it continues in the Golgi apparatus, and the oligosaccharide or the saccharide chain eventually gets completely modified along the way. Dusanka Diuretic has done a yeoman's job of working out the process whereby rhodopsin moves from the rough ER through the Golgi stacks and is shepherded along by G proteins of various sorts. The trafficking part of the rhodopsin molecules on the C terminus, which is cytosolic in its orientation. And then the protein finally arrives at the plasma membrane and goes up the cilium. I've got my my picture of all the people here. There we go. The integral membrane protein is thought to move up the plasma membrane of the cilium out toward its tip. Now, that was questionable for a long time because of the immunogold labeling. Well, what's going on here? And we'll get back to that in a moment. Now, regarding the prosthetic group, the retinoid, where is it attached in the rod photoreceptor? And these look really terrible, these autoradiograms, but that was out of necessity, because when you use vitamin A as a precursor, it gets converted into 11-cis retinal, of course, gets bound to photopigments, but that's not a very stable bond. It's a Schiff's base. And if you drop that retina into fixatives and you dehydrate it, you demobilize, you mobilize the label. So we had to use special methods where we reduced the Schiff-based linkage with BDMA and then extracted extensively with organic solvents. And we saw no label whatsoever in the inner segments of these battered rods in frogs. We know now that 11-cis retinal is required as a chaperone for cone pigment migration in the inner segment to the outer segment. But that is not the case for rods. There are a number of studies that support that observation. We didn't have the foresight to look at cones at this time, so I don't have any images. But in the case of cones, the label should in fact be in the Golgi apparatus or somewhere in the inner segment as well. Now, a huge surprise came to us when we looked at this process more closely over time. When we injected mice in the dark, in their tail veins, with the rest of the body totally enclosed in darkness, we saw that the initial incorporation of the 11-cis retinal was diffuse throughout the outer segment. And then as the label was allowed to accumulate in time, it did form a band of migration, a migrating band, but it was not a really sharp band like one sees with tritiated amino acids. And the interpretation of that is that the chromophore is exchanging in the dark. And it exchanges at an extraordinarily slow rate, at approximately 1.77 times 10 to the 3 per minute per rod. That sounds like a lot, but if you recognize the fact that there are 10 to the 7th per rod outer segment, this would actually take months for the entire chromophore to exchange in the dark. King Wayao subsequently, with his group, has shown that this, in fact, happens in cone outer segments, and he was able to measure that electrophysiologically, although it happens more rapidly in cones than it does in rods. Now, the concept of entry through a receptor into the retinal pigment epithelium by the retinoid was very controversial. Heller and I felt that you had to have a way to control entry of retinoid into the RPE, but others claimed that that was unnecessary. That could simply happen by diffusion from lipid to lipid without the intervention of a receptor. And we did show, provide considerable evidence using iodinated retinol binding protein, injecting it into rats, and then flushing out the radioactive blood, or incubating isolated bovine RPE cells in radioactive retinol binding protein. We saw this very intense label on the basal surface of the RPE, not the apical surface. And all these autoradiograms were done at the time by my technician, who actually spent 48 years total in my lab before I retired, Marcia Lloyd. Along came Hui Sun from Jeremy Nathan's lab in 2003. And he came to me and said that he believed that there was a receptor on the RPE based on our morphological studies and our competition studies and so forth with Heller, and that he would like to see if he could clone the gene for that. Up until that point, I was told by some of the leaders in the field that they wouldn't believe it unless it was cloned. So I was very grateful that Hui offered to take this on. This is an antibody to the protein that resulted from that study, and it's called STRA6, which means stimulated by retinoic acid number 6, something that Pierre Chambon had discovered. He didn't know what it was doing. He put it on his shelf, and it was there, a sequence was there in the database, and you can see how nicely antibodies to it decorate the basal lateral surface of the RPE. The red label is pyropsin, another protein that Hui Sun discovered when he was with Jeremy Nathans. So STRA6, in my simplistic way of thinking, would work something like this. We got retinol binding protein bound to transthyretin circulating in the blood as a complex carrying all trans retinol. We know that that complex leaves the choriocapillaris and binds to the basolateral surface of the RPE. Now, the receptor in the plasma membrane, I thought, could simply accept that transretinol into the cytoplasm of the RPE. We knew that RBP is a lipocalin. We knew that CRBP is a lipocalin. That's the cytosolic retinoid binding protein, retinol binding protein. If you look at the retinoid, the beta ionone ring is here, the tail is here, the conjugated double bond chain, sort of like a rat sitting in a hole with its head deep in the hole. And RBP could simply deliver that to CRBP, and the tail would now go in first, as we knew it was to be, that it was based on structural analysis of CRBP, and that would be the way RBP delivers the retinoid to the CRBP. Well, it didn't turn out to be the case. This, as you can see, is a higher resolution image of the receptor. It's a 9-pass, very hydrophobic molecule, does have a channel through it. And here's the RBP with its retinoid, and we know that the retinoid goes into that channel. But Hui could find absolutely zero evidence to suggest that CRP was the immediate receptor of that retinoid. Subsequently, a group has done electron microscopic analysis cryo-electron microscopy of the zebrafish receptor. And they have found that, yes, indeed, there's a pore there that the retinoid could go through, but a likely pathway would be for the retinoid to travel sideways like this through an opening in the side of the protein and then into the bilayer. And from there, somehow, it could find its way to CRBP. Chris Balchevsky and others have shown that LRAT facilitates the delivery by STRA6 to CRBP. So we don't quite understand how all of this happens. We know if we knock out LRAT in the mouse, that the levels of CRBP drop, the levels of RPE65 drop. So they seem to be sort of interacting with one another, but we don't know the details yet of this delivery. The other interesting evidence is that it seems to be a dimer in the lipid bilayer, the STRA6. That resolution is at 3.9 angstroms, as I recall. Now, we were interested in disrupting this gene, of course. When it's perturbed in human beings, you find a very, very serious polysystem syndrome. People with hardly any eyeballs, multi-system effects. And so we thought we would find a profound effect on mice if we knocked it out. Well, before that happened, Chris and von Lintig using morpholino technology, knocked it down in zebrafish and showed that indeed there were multi-system issues. The eyes were smaller. You can see here the wild type eye, and here's the affected eye. And all kinds of things kind of mimicking what happens in humans. But when we knocked it out in the mouse, my technician Alberto Ruiz did this in collaboration with Norbert Gieselink in Chambone's lab. When we knocked it out, you see here the base of the RPE is in wild type is labeled with STRA6. In the knockout, there's no label. Really, the retina looked quite normal. The outer segments were shorter, but the cell number was normal. And you can see here in this spidergram, the measurements of the outer segment length. So that wasn't very impressive. Steve Nasinowitz, my colleague at Jules Stein, did the electrophysiology. And although the sensitivity was down for the B wave and the sensitivity was down for the A wave, the kinetics were really normal. So that wasn't a particularly striking effect. One very striking effect, though, was the fact that the cones died. So here you see the wild type cones labeled. Here you see STRA6 knockout labeled after multiple months of life. And like IRBP knockouts, like RPE65 knockouts, and now STRA6 knockouts, the cells that really suffer the most are the cones. And that makes sense because we know that retinoid is needed as a chaperone for cone opsins to get to their final destination. One other peculiarity, and this is something that Gieselink had already discovered, but he actually missed it in the initial histology. Steve Nasinowitz picked it up by doing non-invasive imaging methods of the fundus, and he found that there was something blocking the light path. And sure enough, was a persisting hyperplastic primary vitreous, shown here heavily pigmented, here. Virtually every animal had a persisting hyperplastic vitreous. And that's because dissolution of that during development requires retinoic acid. So there was some deficiency there. So we went on to do a bunch of other things regarding the retinoid cycle. I will leave it at this point, but one of the key tools that we developed, starting with Bruce Pfeffer, who was a postdoc, continuing on with Jane Hu, who is still at the Eye Institute, now working with Roxana. We not only studied retinoid transport in cultured RPE, putting whole RBP on the basal side, apical IRBP on the apical side, which is required for efficient release of 11-cis retinal, and we could reproduce the entire visual cycle in a culture dish. Moreover, we were able to feed these cells with Roxana outer segments from Stargardt knockout mice, which are filled with toxic bisretinoids, feeding those outer segments, comparing them to control outer segments, and measuring the inappropriate response by these cells with respect to complement activation. If you give them Stargardt outer segments, they really turn on their defense mechanism in the context of complement. They literally coat themselves with an armor of negative complement regulators. And I know Roxana has given a seminar at UC Irvine on this subject, so I will leave it at that. What are some unresolved issues? Well, the delivery of all-trans retinol via the receptor, how does it really get to CRBP? We really don't know the details of that. We know now that CRBP ultimately delivers retinoid to LRAT. LRAT esterifies it. We know now that the isomeral hydrolase, through our work with Mike Redmond knocking this out, and through the work of Gabe Travis, Ma at Oklahoma, and Redmond at the NEI, we know now that the isomeral hydrolase, as it was once called, is in fact RPE65. We still don't know how the vitamin gets from CRALB, which ultimately carries the 11-cis retinaldehyde. How does it get across to IRBP? Is there a receptor on the photoreceptor for IRBP? And indeed, is there a receptor on the apical surface of the RPE for the IRBP? Because this visual cycle has to run efficiently and in a controlled manner. Outer segment disc biogenesis. This has became a rather hot topic a few years ago, and I'll explain why in just a moment. At the 2019 FASEB meeting, there were still 22 posters and platform sessions on this subject. So it's one that's had staying power, and there's been some really neat work that's been done by a number of laboratories. One question has been, does the opsin, the rhodopsin, actually in a rod it would be opsin at this point, does it actually pass by translational diffusion, or by transport actually, aided by motors, up the plasma membrane of the cilium? Or does it go through the bore of the cilium? Are there vesicles in the cilium? This became a hot topic a few years ago. A recent paper from Futter's lab with her, I guess, probably her collaborator student Burgoyne, has given very convincing evidence that yes, indeed, there is opsin in the ciliary plasma membrane, using both 1D4 from Bob Mulday and RET-P1, which recognizes the N-terminus. There's clearly labeling of the plasma membrane and virtually no evidence of anything going on in the core of the cilium in the axonemal area with respect to opsin transport. A wonderful group of investigators have studied the whole process of ciliary traffic. Joe Bashars, David Williams, Vadim, and others. And the story is developing that microtubules, which are oriented with their plus N here and their minus N here, serve as railroad tracks for cytoplasmic dynein, which then carries vesicles containing rhodopsin to the base of the cilium, and then intraflagellar transport particles cooperate in the transfer from this point to this point, and along the way, the cytoplasmic dynein hands off the cargo to kinesin, which migrates to plus ends. You can see the polarity of the microtubules changes at this point. And so the transport then is upward. It's sort of like a horse pulling a barge in a canal. And the canal is the plasma membrane, and the horses are here. And finally, at this point, the transport particle comes off, intraflagellar transport particle comes off, hops back on again and carries other things back in this direction with dynein that is negatively directed. So that's a lot of stuff going on. You also have the Bardet beetle some, the BB-some, playing a role here. And all of this is reviewed very nicely in photoreceptor cilia and retinal ciliopathies. There are just scores of genes that cause ciliopathies. And of course, they have dramatically negative effects on photoreceptor health. So here's a nice review for you to look at if you're interested. Now, a few years ago, a group at Cornell, using what they thought was a sort of novel fixative, acrolein, suggested that discs did not form by evaginations, as we were classically taught, but in fact, by migration of vesicles up the core of the cilium and coalescence of those vesicles into discs. And the acrolein, from my perspective at that time, was not a good fixative. A lot of us had tried it in our youth. It's not a dialdehyde, unlike glutaraldehyde, which is a nice cross-linker with amino groups. It's actually one of the major ingredients in tear gas. And it is not, in my view, a good fixative. So here's a review by Imanishi and his group, showing a cartoon illustrating these two vesicle coalescence models versus growth from in a lamellar fashion. If you went back in the literature, for example, this paper by Dick Young years ago, using classic fixation, glutaraldehyde formaldehyde mixtures, perfusion of the animal, you can see beautiful evaginating discs here, the basal one, and then they get bigger and bigger, then they pinch off. These data were in the literature, and from my view at the time, the second law of thermodynamics would argue against any process whereby fixation could cause an artifact like this. The second law says that it takes energy to impose order on a system. And I don't see how you could impose order on a system like this just by infusion, and I didn't see how you could create an artifact like that. So subsequently, Burgoyne et al., Ding et al., Bolin et al., and so these are from Footer's lab, Arshavsky's lab, David Williams' lab, using really elegant morphological methods, have combined to come up with electron microscope tomographic images and other images to show these beautiful evaginations. So I think the consensus now is that the original model proposed by Steinberg and Miller is correct. Now, a major contribution to our understanding of outer segment disc morphogenesis has come from the discovery of peripherin 2. Gabe Travis and Rick Sutcliffe pointed out that there was a gene in RD2 mice that was responsible for the spontaneous absence of outer segments in those mice. Mice that were originally described by Soma Sanyal in the Netherlands. Bob Molday actually discovered the gene product and called it peripherin. That name had to be changed to peripherin 2 because there was already a peripherin in the literature, one that found in myelinated axons. So we call it peripherin 2 now. And the phenotype is thus: in the RDS minus animal, or the peripherin 2 minus animal, there are no outer segments. They're just inner segments. In the heterozygote, you have burritos or jelly rolls for outer segments. And the question is, what is the cause of all this? Well, Neer and Papermaster, and later Usakura and myself, about a year later, showed that the tips of these cilia, and these are really primary cilia like you find in many other cells in the body, the tips of the cilia do have opsin in them. And in the extracellular space, as had already been described by Sanyal and others, there are all these vesicles. And those vesicles are labeled with opsin antibodies. So they're full of photopigment. Well, what's going on here? We found that we could completely cure this phenotype transgenically, Travis Sutcliffe and myself, by transgenesis, injecting a normal gene into the ovum of mice, that you could form totally normal outer segments. And interestingly, if the rescue transgene fell on an X chromosome, you had X chromosome inactivation, of course, as we well know it. And wherever you had non-rescue, you still had cilia budding off their little bubbles. And wherever you had the transgene, rescue transgene and the photoreceptor, the rods were normal. So this was a nice example of making an X-linked animal, if you will. So what is the function of the peripherin? Well, it has multiple functions, turns out. Vadim Arshavsky's group has shown that the absence of peripherin 2 allows the cilium to form ectosomes. So what's an ectosome? You've probably heard of exosomes. Exosomes are formed when you have endocytosis of extracellular material and membrane to form multivesicular bodies. And those in turn, that material in turn can be secreted again out into the extracellular space. An ectosome is formed by machinery right under the surface of the plasma membrane, and it's a budding process. And this is indeed what you have in the RDS mouse. You have all those little bubbles that have opsin in them are ectosomes. And this is a normal process. This is how cells normally communicate with one another. The ectosomes are produced and then they're taken up by another cell. They can have RNA in them. They can have all kinds of goodies that another cell would want to use. Another cell will take it up again by this endocytic process. And Vadim showed elegantly that if you look very closely at a cilium, you can find the little guys budding off the surface right there. So his conclusion was, or his group's conclusion was, if you have no peripherin, you blow bubbles. If you add peripherin, you have beautifully organized discs. We know that the peripherin is in the hairpin loops of the rod and cone discs. This part of the disc does not have peripherin, and therefore it doesn't have these hairpins. If you want to read a really nice historical article with fairly current data in it, go to this thing this article in Progress in Retinal Research. This is the review article is written by Moon and Ash's group. And then there's another paper from Palchevsky's group that also does a functional analysis of peripherin and its counterpart, its companion called ROM1, which is also a tetraspanin, and how they interact with one another covalently to form this rigid, beautiful curvature on the edges of rod and cone discs. Now, are there other proteins that provide structural support for outer segment discs? We knocked out STRA6 with Travis and did ultra rapid freezing and freeze substitution analysis, and found that in the total absence of ABCA4, there's really very little perturbation of the hairpin loops in the rod discs. So that protein, although it's a big, important protein in the context of Stargardt disease, really doesn't provide much in the way of structural support. We had to do diffraction analysis to show that there were any differences whatsoever in the ordered nature of these disc edges. We never published this because we didn't really know what to do with it at the time. Probably should have put it out there somewhere. Wayne Hubble has proposed that opsin can carry out homotypic interactions in the space within a disk, such that two opsin molecules from opposing membranes can interact with one another. So that's a possibility for some structural report. It's first proposed by Steve Fleisler, actually. And then Vadim Arshavsky has recently shown that the progressive rod cone disease gene is actively engaged in the flattening of discs. And I believe Uwe, what's his last name, in Germany, first name Uwe, has provided some immunogold evidence for the localization of this, as has Vadim near the base of the outer segment.
Wolfram.
Uwe Wolfram, thank you very much. Okay, so what else do we have? We've got prominent, which is on this part of the disc, the open part of the cone disc, the open part of the rod discs. Prominent is another protein that provides structural integrity. And the list kind of goes on. We have interaction between this gated channels and peripherin. I'll show you some EM evidence of that as well. And other components like actin. The role of actin here is still unresolved. We think that might be involved in the growth of discs in some way through polymerization, but that hasn't been completely resolved. We know it's there. This is work from a postdoc of mine, former postdoc, who you all know, I'm sure, Mike Chaitin. He showed that there's actin here where the discs are forming. With Beth Burnside, he decorated that actin with heavy meromycin to show it's filamentous. Here you see it in the frog. And here, if you use cytochalasin, Dave Williams and Steve Fisher, which is a disruptor of filamentous actin, the discs overgrow. So it seems to be involved somehow in disc membrane formation. These little linkers in the frog between the necks of the hairpin loops have not been resolved. You don't see them in mice, as far as I know. I've never seen them in a mouse, but we don't know what's causing that either. Anyway, it seems to form structural support. Now, this gets really interesting, this business of phagocytosis and how it's controlled. Matt LaVale, after we published our original paper in 1969 on disc shedding, was looking at rat tissue from various times of the day, and he noticed that at certain times of the day, there were virtually no phagosomes in the PE, and at other times of the day, there was an abundance of phagosomes. And he published this in Science and provided evidence that disc shedding in the rat, and it turns out to be the case in the mouse and other mammals, is circadian. And that really opened up a whole new field. Within, I think, 2 years or a year, Arvo had 35 papers on disc shedding, 35 platform presentations on disc shedding. In the leopard frog, the grass frog, Rhinopipians, it's not circadian. If you don't turn the light on, they don't shed. But if you turn the light on, you get a burst of shedding. So not all species are the same. Xenopus is sort of an intermediate, sort of a blend of circadian and non-circadian, as Bashars has reported. Now, this very interesting experiment showed how the retinal pigment epithelium cooperates with the photoreceptors in shedding. If the two are in contact with one another, and if you have labeled phagosomes, now I think this was probably Xenopus, but I'm not sure. You have a nice shedding response. Pigment epithelium is taking up these radioactive packets. If you pull off the pigment epithelium, they don't shed. If you pull the pigment epithelium part way off, the part that's in contact has a nice engulfment response, but where there is no contact, the outer segments don't shed. So clearly there's some cooperative process going on. You need RPE and photoreceptors working together. One misconception about shedding involves the microvilli of the pigment epithelium. It's not the microvilli that do the eating. It's transient Lamellapodia that form in response to all the signaling that goes on during shedding. They extend themselves downward and they literally mechanically dissect the tip of the outer segment. And Bashars and Matsumoto and Defoe showed that there's actually a transient opening to the extracellular space using Lucifer yellow. I'll show you that in a moment. And Curry, Hollyfield, and Rayborn showed that in the frog, you actually have vesiculation of discs if they are, if you catch them at the right time during the shedding response. Now, here's further evidence about these Lamellapodia. We deliberately cultured cells that have no apical microvilli. That's not hard to do if you got your culture conditions wrong. And here you have one phagosome that's inside the cell already. Here you have a great big one that's probably too big to eat all by itself. But this big old Lamellapodium has leapt out and grabbed onto that thing and is trying to pull it in without a single microvillus to be seen. An incredibly important animal model for our understanding of phagocytosis has been the RCS or RDY rat. We initially, with Mike Hall, showed the phagocytic defect, and then Matt LaVale, in an even more elegant set of experiments, showed that it was really the RPE that was to blame, along with Doug Volroth. Matt had this incredible collection of isogenic rats, both control and RDY, that were incredibly, really essential for Doug Volroth to do the genetics, the positional cloning. And so just to remind you, in a wild type rat, this is what it looks like. You got a band of radioactivity, packets get shed, RPE digests it. In the RCS rat, that process is not taking place. All this CRETS builds up in the subretinal space, and there is virtually no phagocytosis. So it turns out that the gene for that is called MERTK. It's a kinase. And here's the super elegant experiment where Matt and Mullen at Utah made tetraparental rats. They mixed the blastulae of a normal rat with an RCS rat, and the RCS rat has a non-pigmented or lightly pigmented epithelium. The wild type has a pigmented epithelium. Wherever you have wild type pigment epithelium, got normal outer segments. Whether you have mutant pigment epithelium, there's no eating. So that was a beautiful, and cell death underneath it. Beautiful example of where the mutation is being expressed. And then, of course, there are a bunch of labs, including my own, that transplanted wild type RPE into the subretinal space on top of the mutant RPE. And when you do that, the outer segments, the photoreceptors underlying the wild type RPE are normal. Now, what is the mechanism for all of this? And I really tip my hat to Sylvia Fenneman's group now at Fordham University for shedding a tremendous amount of light on this subject. MERTK was absolutely essential to understanding, of course, but there are a lot of other players. We have this process called phospholipid flip-flop. It's first described by Hardin McConnell, Wayne Hubbell's mentor at Stanford. Normally, the phospholipids in the bilayer are asymmetrically disposed. Phosphatidylserine is largely on the cytoplasmic leaflet and not on this extracellular leaflet. And during certain processes like apoptosis, there's a flip-flopping of the phosphatidylserine, a flip-flop from the cytosolic leaflet to the extracellular leaflet. And that can be accomplished in two ways. We have enzymes called scramblases that can scramble the whole thing up and make it more symmetrical. Or we also have inwardly directed ABC ATP binding cassette proteins that use ATP to maintain this asymmetry. So if you inhibit that inward directed maintenance process, then you could also have inappropriate localization of PS on the extracellular surface. And a reagent that reports when that happens is called annexin 5, annexin 5. So here's an absolutely gorgeous experiment. I wish I had done it. Here's Matsumoto and Bouchard's is evidence that you have transient opening of discs near the tip in the frog, suggesting that something is going on preparatory to shedding. This is an experiment done by Feneman's group and in collaboration with Ralph Langen. Ralph is the husband of Jeannie Chen, and he developed a derivative of an exon that glows, that fluoresces when it binds to PS. So if there's no PS there, there's no glow. If you've got PS there, the tips of the outer segments glow, and look at them glow. These animals were entrained to the light-dark cycle. They were ripped out of the eye and exposed to this reagent, and the tips are just glowing with this bound reporter, this fluorescent annexin-derived reporter that Ralph prepared for the study of apoptosis. Here is an animal, a series of animals where an integrin on the apical surface of the RPE, A5 beta 5, is knocked out. When that happens, there is no circadian labeling. The pattern is similar at 1 hour, 0 hours, 1 plus, +7. There's just this sort of low-grade shedding in phagocytosis that goes on in the A5 beta 5 knockout. Whereas in the wild type, you have this peak after light onset, and that peak is circadian in nature. Okay, so let's try to put all of this together. Largely the work, again, of Feneman's group. So let's start here at the plasma membrane of the outer segment. You've got asymmetrically disposed PS on the cytoplasmic leaflet. Something happens, some kind of signal comes along, and you have scrambling of this bilayer in some fashion by some mechanism we don't understand. In the extracellular space, in the subretinal space, is milk fat globule E8. That recognizes phosphatidylserine, either regular PS or oxidized PS. And again, we don't know the chemistry of the PS oxidation. Those oxygens can be added all over the place depending on where the double bonds are. So this MFG opsonizes, binds to this integrin, this alpha 5 beta 5, on the plasma membrane of the RPE. Now, if this is missing, you don't have synchronous shedding at light onset. If it's present, you have synchronous shedding. That, in turn, when it's activated, activates a G protein, RAC1, and that is involved in the assembly of these pseudopods, these F-actin-rich pseudopods that engulf around, that surround outer segment tips. And then, through the activation of focal kinase, which is also involved in cell adhesion, you get the activation of MERTK, and Gas6 protein S also has recognition sites for PS, phosphatidylserine. It opsonizes MERTK, and MERTK is involved in the engulfment step. The flip-flop PS is the eat me signal. Then after that eat me signal appears, all of this other stuff happens. In addition to that, there's a break, as Feneman has shown. You've got semaphorin 4D here, binding to plexin B1 in the dark, and somehow during the circadian entrainment process, or when light comes on, apparently the semaphorin D gets degraded. And now the brake is off. And when the brake is off, then all of this stuff starts to happen, this formation of the pseudopods. Mike Chayton and Mike Hall years ago showed that if you plop some outer segment fragments on the pigment epithelium, you have these little pseudopods surrounding those things. And that is what we saw in the previous cartoon. That's what happens when you activate actin via that G protein. So it forms these, what they call the phagocytic cups. All right, digestion of ingested outer segments. I won't dwell on this. I'll just say that David Williams and others have shown that myosin 7A is involved in this, that the phagosome that's taken up has to be moved from the apical region of the pigment epithelium into deeper regions of the cell. And in Usher models, Usher syndrome that involves myosin 7A, that process doesn't take place, and therefore digestion is not efficient. Digestion is carried on by various cathepsins and other molecules. And years ago, we showed that the phagosome in the pigment epithelium, using rapid freezing methods, we showed that the phagosome actually kisses the lysosome, or the lysosome kisses the phagosome, and you have little channels that form transiently between the lysosome and the phagosome. This is cathepsin D labeling here. David Williams' group has now gone on to produce movies of this process where the phagosome and the lysosome are frantically rushing around within the cytoplasm. They bump into each other, they kiss and they run, and they kiss and they're not just a single romantic event. They kiss and run, kiss and run, kiss and run until enough stuff is injected to digest the outer segment. So this process of phagocytosis is important, we think, because the cells live in such a toxic oxygen environment. They need to be turned over to stay healthy, and so that's good. However, in diseases like Stargardt 1 and certain forms of age-related macular degeneration, the bisretinoid toxic products, which Chris has shown can actually diffuse from the outer segment into the RPE, are also engulfed along with these packets, and they can lead to inappropriate activation of complement and compromise important vital processes. Here's the chemistry of that process. Phosphatidylethanolamine gets inappropriately linked to two retinoids if the retinoid doesn't get pumped out of the discs fast enough. And you have this A2E molecule, which in itself is dangerous enough, but then through singlet oxygen, it gets converted through oxidation into a multitude of things that become more water soluble. They can roam throughout the cell, they can leave the cell, activate complement, and cause a lot of problems. So we bid you adieu. This is Jane, who worked 30 years for me, culturing RPE cells. She now works for Roxana, with Roxana. Alberto did the cloning of LRAT and the Straw6 knockout and a bunch of other things. He was our molecular biologist. He lives retired now in Irvine. Marcia, 48 years with me, EM technician. And these are some core technicians who we enjoyed working with very much, Shannon and Steve. We took this picture before the pandemic came. University had a new requirement that we wear goggles in the lab during certain procedures, and we didn't anticipate the restrictions that would follow that. But nonetheless, it's been 50 plus years of a lot of fun, and I thank you for joining me this morning.
Ian, thank you so much. Anybody who has a question, please drop me a note in chat that you would like to ask question, Dr. Bock. Dean, I have one conceptual question related to phagocytosis. So at some point, you pinch off a number of discs that they get phagocytized, and either you have a fusion of disc with plasma membrane at the tip, not to allow holes in the cell. Otherwise, you will have a rash of calcium and most likely you will kill the cell. How do you imagine that process? What are your thoughts about this process of pinching off and opening the cell? Because there's no any other way you will get them out.
Right. Well, this process of that, the apoptosis people call efferocytosis is specifically designed to avoid that. The whole process of rapid phagocytosis is designed to take dying cells or pieces of cells and quickly internalize them into the macrophage, and in this case, the RPE. So the process is quite rapid, and the whole process of packet isolation and resealing of that packet is designed to prevent that leakage of material out of the packet. If you just isolate outer segments, and work with them in a test tube, this flip-flopping takes place, even though it's no longer under this circadian control. So that's why people are so successful. I used to think, well, why is it that RPE cells eat under the right conditions, eat these outer segment fragments so efficiently? And Sylvia has shown you've got to have milk, fat, globule, protein there. You have to have all the right stuff. You can't just throw outer segments on an RPE and get rapid phagocytosis. But if everything is right there, it happens very quickly.
Well, the quickly cannot be faster than diffusion, right? And calcium will go with the rate of diffusion. So quickly is kind of relative. If in terms of the diffusion, this is a slow process.
Well, you know, there's a lot of, I can't answer your question, obviously. There are a lot of things happening out there that we still do not understand. There's a new paper out of Germany, a group of people who are interested in LRAT mutants, I believe. At any rate, they're making iPS cells, and they show that when you put an outer segment on a cultured RPE cell that's too big, these pseudopods rapidly fragment it into smaller packets, like they're furiously trying to keep them from leaking. Mm-hmm. But like you say, fast is relative. So, and calcium leakage.
Yeah, it is a puzzling, because one of the mechanisms could be that you have at the tip, as it starts breaking off, a fusion of the part of the disc with plasma membrane at the tip.
I think that has to be the case. Yes.
Let's move to. I will throw at you Steve Fleisler. Would that be acceptable?
Yes. Bring him on. Where is he? Steve?
Steve, are you there?
Yes, I am. I was still muted. I'm sorry. So, Dean, just elegant as usual. My question to you is, it's been long observed that the opsin that goes up to the outer segment doesn't seem to come back to the inner segment under normal conditions. But if you have a genetic disease, particularly a photoreceptor specific genetic disease, now you see the opsin comes back flowing around the plasma membrane of the inner segment, even to the synaptic end. What is the normal traffic cop for that prevents that lack of vectorial absolute directionality?
Well, the normal traffic director is the microtubule and the molecular motor that schleps the vesicle in the right direction. The microtubules are oriented, as you know, plus minus, and so the direction is forced that way. However, when you have a really sick photoreceptor and you see this randomization of label in the plasma membrane, it's not clear to me whether this is backflow per se or whether you have inappropriate fusion of the vesicle in the first place with the plasma membrane of the inner segment. So I'm not sure whether it's backflow or whether it's never getting to the right destination in the first place.
Thanks, Steve.
Yeah.
Any other question?
I have a question.
All right.
Dorota here. It was an amazing lecture, and thank you for putting everything in the context, Dr. Bach. And I actually am a little bit interested in the end of the process. So how do you think the phagocytosis is stopped? Why RPE is not eating more? And is it only circadian rhythm that regulates that process? I think it is really fascinating.
Well, the eat me signal is necessary. If you don't have an eat me signal, you won't have phagocytosis. So somehow there's a difference between a mouse or rat and a frog in that respect. What is it that triggers the eat me signal? What causes the scrambling of the PS to take place? And obviously, it's the signal must be more complex, I would think, in a mammal than it is in a frog. But if there's no eat me signal, there's no phagocytosis.
Well, this is a little bit different from macrophages, for example. They don't need to have whole cell covered with the eat me signal to eat whole cell. That's why I think there's
Right.
some process to stop like eating frantically the whole cell.
That's right. When I used to teach a little immunology in my cell biology course, I called macrophages the goats of the immune system. They'll eat tin cans and radishes and all kinds of things. The pigment epithelium is more specific in that regard. And again, it must have something to do with ligands on the surface. Macrophage must be much less specific in terms of the ligands that it will recognize. Sam Silverstein used to call it a shark feeding frenzy. When a macrophage encounters a red blood cell, it just goes wild. And there's this zippering effect that takes place between the plasma membrane and the effete red blood cell. And it gets quickly internalized. So obviously the machinery is different. The pellet of the pigment epithelium is much more refined, I would say. But, you know, that's a kind of a flippant answer. It means I don't know the answer.
And I have one quick question. I noticed you didn't mention CD36
Yes.
in your talk. And actually, this is a very interesting protein. So would you mind mentioning and
Yes.
letting us know what you think?
It's an interesting molecule in many respects. It actually was the first one that was implicated in the RCS rat by Sandra Riom and Janet Sparrow and Silverstein. They suggested that CD36 might be the key receptor involved in the RCS rat. It turned out not to be the case. It's MERTK, but it's everything else too. Obviously, alpha 5, beta 5 is important. And CD36, from my reading, recognizes oxidized PS more specifically.
I think it depends when the literature goes. It also is the lipid transporter, actually fatty acid transporters in other literature in totally different aspects. And that's really interesting because it may solubilize the membrane.
Right.
Oh, yeah.
Yeah, so I think it's been a little bit ignored. The excitement over MERTK has been very high, and the integrin and all that, but certainly it's worth taking a stronger look at CD36 as well.
Thank you for your amazing talk.
Okay, thank you.
All right, before we go to Vadim with his question, I just wanted Dean to stay after the presentation and read all of the comments in chat. There are so many complimentary remarks, so you will be very pleased. But now we're moving to Vadim, and then we will go to Aisha, and then we go back to Steve Fliesler, who is anxious to ask you another question.
Dean, thank you very much. That was a fabulous presentation. My question is about as general as it gets. So if there's a single question regarding the entire process of outer segment formation, turnover, anything, for which you would like to have an answer, what would be that question?
Rephrase it for me one
more.
Like
what is the remaining unknown about outer segment's life?
Oh.
Kind of your most favorite question. Like if you went back to the lab, you know, if there was a single question you would like to address and you had all the tools, what would be the question?
Well, I think some of the unresolved questions regarding signaling at the tip of the photoreceptor. What is it that signals this scrambling to take place? What are the predecessors to that? I don't think we have very much insight. It probably involves calcium, which would make Gordon Fain happy, I'm sure. But that is one area I think that really needs to be explored. And also, I'm intrigued by this difference between rods and cones. How does the cone stay conical? Now, I know that in the foveola, the outer segments are not very conical, if at all. And there's still argument, and I think you might be able to answer that with your fixation techniques for showing open discs. Are all the cone discs really open in the foveola, where there are 35,000 red and green cone outer segments? It's a tough nut to crack, because I think you need to use perfusion methods, as you have in your studies and the others as well. Pulling off the retina is very traumatic. We find if you peel off the retina, vesiculation happens very quickly. Vesicles are the low energy state of a membrane. And when Usakura ripped off mouse retinas and slam froze them and did all that beautiful morphology and saw all these vesicles, the problem was he was creating an artifact before he rapidly froze the outer segments. So it's technologically difficult, but I think it would be nice to see some studies done on a rhesus monkey or something, or a very short post-mortem human eye. I think people agree now that poking holes in rat eyes before immersion fixation is not a good idea. You should immerse the whole globe. Just leave it alone and immerse it. We used to think you had to have a pathway inside for the fixative, but you're actually causing problems doing that. But I think those are some of the things as a cell biologist that I would like to explore if I were still exploring.
Thanks.
Isha.
Hi.
Hello.
Very nice presentation. I really enjoy it.
Thank you.
So if the phagocytosis, it's a very mainly ordered mechanism, and the RPE only, it's involved in the phagocytosis in the tip of the outer segment. Why in some cases, like I will talk about my experience when I was working with the mice that have a problem with the lipids, and specifically with cholesterol. Why in some cases we see that the RPE is overwhelmed, and when we do the electron microscopy, we can see that now RPE it's working in phagocytosis, but then it's calling on whether it's macrophages or microglia or something to help with the cleaning. So you think there is a talking between the two, the RPE saying that, oh, this is too much coming in. I need the help from maybe microglia from inner retina, or from myeloid cells coming from the general circulation.
Well, certainly in the case of the RCS rat, when the retinal pigment epithelium cannot do its job because of this mutation, the microglia migrate upward and help to clean up that debris. Also, even in normal animals like the goldfish, Dick Young and Bill O'Day showed years ago that there are macrophages roaming around in the subretinal space that literally drape themselves over the tips of cone outer segments and assist in the phagocytosis. So if you have a really traumatic injury like heavy light damage to photoreceptors, there's a massive uptake of material into the pigment epithelium, as Kuwabar and Friedman showed way back in the 60s. So you can overwhelm the system, and you certainly there are cases where help is needed by other cells.
Without RPE call, without any, without any mechanism or pathway, or maybe something from the RPE.
Well, it would be signaling.
Like pure immune, mechanism.
I think it's signaling that's created by the presence of the debris, indicating that there's some, it's like a fire alarm calling the microglia in and other cells, other phagocytic cells. We often think of the RPE as the champion of phagocytosis because it's really post-mitotic generally, and it has to function throughout life. So you can find 100-year-old people who have perfectly healthy retinas with still a functioning RPE. The closest thing I can think of that comes to an RPE cell is the Sertoli cell of the testis. And the Sertoli cell of this testis, literally, it's like a Mueller cell in the neurosensory retina. It literally surrounds everything. It's got this massive cytoplasm, and all the spermatocytes and the developing spermatids are embedded, not inside the cell, but enveloped by the cell. And as each sperm cell matures, it casts off most of its cytoplasm, and the Sertoli cell eats it. The Sertoli cell is also the cell that forms the blood testicular barrier. And so from the time a boy reaches puberty and starts producing sperm cells until many, many decades later, these same Sertoli cells are eating all the, I mean, millions, hundreds of millions of cytoplasmic chunks that it has to eat every week.
Thank you so much.
Yeah.
Let's move to a beautiful Steve Flisler.
Thank you. So Dean, that mention about the possible homotypic binding interactions between opposing intradiscal leaflets of opsin, that process was at least postulated to require N-glycosylation of the opsin, because if you prevent glycosylation, you don't get that close apposition of the discs. But if you trim the glycan, so you still have a glycan, it's just not the mature form, that doesn't perturb the process. So those two pieces of evidence would argue that you actually require the glycan to get that close apposition of discs.
Yeah, in Hubble's model, he used structural data to match the homotypic interaction. He didn't, I don't think he said a whole lot about the glycans, but he does include them in his model. He has them there in that model if you look at that review. So right. I don't know if he was ignoring the glycan or not. I assume that was part of the structural analysis that he did.
I guess the point is that the simply having the polypeptide is necessary but insufficient to have.
Right. Yeah. Well,
the problem with this model would be, Dean, that it will immobilize rhodopsin on both sides. And again, it will go against the idea that there is some mobility of rhodopsin. So it's on one hand, he thinks it's so mobile. On another hand, it is so immobilized because of that interaction.
Yeah,
it just doesn't jive together, in my opinion.
So it's not covalent bond forces.
No.
We're talking about hydrogen bonding.
And there were individual mutation, as you know, Steve, in glycosylation sites.
Yes.
And the mice were made.
Yes.
Good
Danny.
point.
Danny? You're speechless.
Sorry. That was a fantastic presentation, Dr. Bach. I learned a lot from a historical perspective. I want to, my initial question was harkened back to the question prior, which was the unknown in terms of when you have a photoreceptor degenerative state, and also going back to some of what you mentioned regarding how phagocytosis of the RPE is so specific, whereas the macrophage is more like a goat. In the context of a degenerative state, you're now going away from a circadian rhythm effect, where you need the RPE to now eat dying photoreceptors that may not happen just when the light switch is on, right?
Right.
And then you're also eating the RPE is eating potentially a meal that they had never expected, which would add more RPE stress. And curiously, what we found you know, and you'd mentioned in the MERTK knockouts that you have microglia in the subretinal space potentially aiding. And that was shown, you know, with some rhodopsin particles many years ago. I'm blanking on exactly who, and I apologize. But we had shown that if you deplete microglia in the light degeneration setting, that those subretinal microglia in their absence, you get a major RPE defect, where you get RPE dysmorphogenesis, whereas in the presence of those microglia, the RPE looked relatively intact, the morphology. So, and we showed a similar phenomenon in the P23H knock-in mice, Chris's mice that they developed in his lab. The phenotype was a bit different, but the morphology of the RPE was also somewhat deranged, I guess, or dysmorphic to an extent too, suggesting again that these microglia, particularly in the subretinal space, are playing some role in helping the RPE sort of remove debris that it may or may not be able to handle. I don't know if that jives with your thinking.
I think, yes, I think this is a case. You mentioned dysmorphic appearance of the RPE. It's probably because of the toxic materials that are released in the subretinal space in the absence of those microglia. Microglia are not taking that stuff out of commission, and the result is a sick RPE cell.
Thank you.
Mm-hmm.
All right, let's move to Grażyna, and then I will have one question as well. And the final comments will go to Cheryl Kraft. So Cheryl, just get ready. Grażyna.
Yes, thank you, Dr. Buck, for a fantastic talk. It was great.
Thank you.
And I am following on Dr. Saban's question a little bit about the macrophages, because we frequently see them when studying the models of retinal degeneration or impact of the drug on prevention of photoreceptor cells. But have you seen, what is your thinking about the interaction between the macrophages and the RPE? Sometimes there are those holes in the RPE that some people postulated that maybe macrophages left them. So I am just curious about your insight on this subject.
Did you say holes in the RPE?
Yes, pores.
Pores in the RPE. Yeah, we believe, I guess, that there are microglia developed from resident cells that normally live in the inner retina and are mobilized under pathological conditions to migrate north, if you will. But there are other macrophages that enter the retina, I think, from the circulation and help out. So I think we have a multiplicity of helpers that can come in and alleviate pathology. Now, when you mention a pore, could you elaborate a little bit more on what you mean by a pore?
They could almost like a perfect circle sometimes even. So that there will be the size of it approaching sometimes almost the size of the RPE cell.
Right. So this is in an intact animal, histological section, or?
It will be in the intact eye.
In the intact eye. And you see
not fixed, no processing to the eye.
Yeah. Yeah, it's something I have.
It looks like Dean and Grażyna will get together and look at those sections. And that
I'd like to see them.
All right, fantastic.
That would be the best. Thank you.
Yeah. So Dean and Vadim, you guys have the question about the big picture, what is there? And sometimes we do experiments that are left unanswered. I remember working with LRAT mice, and Dean, of course, you have so much experience with those animals as well. And you can charge opsin with artificial chromophore, like Nice is retinal, and take these mice into darkness for half a year. And as you know, in LRAD, there is no retention of vitamin A. Everything will be washed out very quickly within days. And yet, the 9-cis will be recycled for so many cycles, 6, 7 months, you will have this recycling of vitamin A. So once it gets phagocytized, there must be some mechanism by which that phagocytized material, now opsin is degraded, but the chromophore is transported back in the direction of regenerating fresh opsin.
Yeah. Well, I didn't know about the long half-life of 9-cis. That's very interesting. With respect to entry into the RPE, it's believed by Hui-Sun that the STRA6 will allow passage of retinoid in either direction.
That's correct, but then you wash it out, right? In LRAD, you don't have much of retinoids for
Right.
a time.
Yeah. Well, it has to be trapped somehow. Do you know what is relative affinities of 11-cis, the 11 CRELB for 9-cis versus 11-cis?
Well, 11-cis is extremely tight. It's around 10 to minus 10, 9, somewhere on that order, and 9-cis is about 1 order of magnitude lower.
Okay, so that can't be the answer.
It must be some very direct transport, otherwise you will get diffused out. There is a drop in opsin regenerated level, but this drop is about 50%, no more, over half a year. So that's truly amazing to me and unanswered. But
yeah.
All right, we had a fantastic talk, fantastic questions, many wonderful presentations. Let's go to Cheryl. Are you still there?
I am still here. And
fantastic.
It's just wonderful to actually see so many people to enjoy Dr. Bach's great presentation. I think, I really look forward to these Friday mornings, Chris, and you've really put a great group of speakers together. But, I think what Dean has done over this last hour and a half is really, bring everybody up to date. And so covering it from the beginning to the end and seeing the UCLA marching band going across with his original data with Dick Wood is just amazing. And I think we're so fortunate. And I see that he's an emeritus professor now, but I think we could still convince him to come back to the lab and help us. So I think that the overview and just the amazing body of work, not just from Dean's group, from Matt Lavelle, all the way through to Sylvia Feynman. It's just, and all the passages between Gabe Travis and everyone is just, really made this an amazing story that we're still trying to puzzle and understand. So as we finish up, of course, Chris, you did catch me off guard. I would have had a prepared speech, but I've never been short of words, but it's just wonderful. And I just wish everybody the best. We missed everybody at our booth. So next year in Denver, and I hope Dean Bach will be there. So
Dean, just go to a chat room and read all of the other messages there.
Okay,
yes.
Thank you, Chris, and thank you all.
Thank you, Dean.